Hello and welcome to a UK NEQAS for Microbiology webinar on the detection of Carbopenemase-Producing gram-negatives delivered today by Dr. Katie Hopkins. I'm Jennifer Henderson, the scheme organiser for Bacteriology and Mycology here at UK NEQAS, and I will be hosting today's session. For those of you who haven't managed to attend one of our previous webinars, I'll quickly go over a few bits of housekeeping. So as with the previous webinars, we won't be using the chat function to answer questions, and we ask that you don't raise your virtual hands. Our speaker today will present the slides, followed by approximately a 15-minute Q&A session. So if you do have a question to ask today's speaker, please submit it into the Q&A function found on your screen. We will be prioritising the questions which receive the most upvotes. So if you see a question which has been asked and you would like that one answered, please give it a thumbs up. This will upvote the question and move it further up the list. The links to the recording will then be sent to all registered delegates within the next few weeks. If you've missed any of our previous webinars, these recordings are already available on our UK NEQAS for Microbiology website. Just click on the blue webinar tab on our homepage and it will take you to the full list of presentations, which also includes the link to the recording. Our speaker today, Dr. Katie Hopkins, works within Public Health England's Antimicrobial Resistance and Healthcare Associated Infections Reference Unit, which is the national reference laboratory for investigation of antibiotic resistance in and characterization of healthcare associated and sexually transmitted bacterial pathogens, which is based in Colindale in London. She's been the lead clinical scientist for the Antimicrobial Resistance and Mechanism Service since 2018, which undertakes susceptibility testing and molecular detection of resistant determinants of public health importance in a wide range of gram-negative and gram-positive bacteria exhibiting unusual resistance. So that's enough from me. I will now hand over to Katie, and I hope you all enjoy the presentation. Katie, the floor is yours.
Thanks, Jen. So good afternoon, everybody. I'll just try and share my slides. Okay, so hopefully you can all see those slides. So yeah, this afternoon I'm going to talk about detection of Carbopenemase-producing gram-negative bacteria. I'll just show you a quick outline of what I hope to discuss this afternoon. So I'll give you a brief introduction into carbopenemases and why we need to be able to detect carbopenemase producers. I'll then go on to the main part of my talk, which is how we detect carbopenemase producers, and I've broken that down into phenotypic and molecular methods. I'll then highlight some further guidance, which you might find helpful, and then I'm happy to take any questions at the end, either with regards to the talk, or if there's anybody from the UK who has any questions about our reference service, then happy to discuss those as well. So multi-drug resistance is a serious and increasing threat to the treatment of gram-negative infections, and this is particularly relevant to gram-negative bacteria that are resistant to the beta-lactams, and we're seeing an increase globally in such organisms. The table that I've shown here is of the escapee pathogens, and these are the bacteria that cause the majority of healthcare associated infections. We can see that 5 of those 7 organisms are gram-negatives, and in all of those, ESBLs or multi-drug resistance is now an established problem. This means that we've had an increased reliance on the carbapenem antibiotics, so that we can see that carbopenemases are either an established problem or an emerging problem in all of these organisms. Now, one of the problems is that carbopenem resistance is usually combined with resistance to other non-beta-lactam antibiotics, such as the aminoglycosides and the fluoroquinolones, and this leaves us with very few remaining treatment options. And it's got to the point that the WHO have highlighted Carbopenem-resistant Enterobacterales, Acinetobacter, and Pseudomonas as critical priority pathogens for which new antibiotics need to be developed. So the maps that I'm showing here are ECDC EARSnet data, and at the top right there we've got, the top left we've got E. coli, and then top right we've got Klebsiella pneumoniae. Bottom left is Acinetobacter species, and then bottom right is Pseudomonas aeruginosa. And each country within Europe is coloured according to the prevalence of carbapenem-resistant invasive isolates of that particular bacterial genus. And the more warm the colour, the higher the prevalence of resistance. So if we look in E. coli, most European countries, in fact, all of them are reporting carbapenem resistance at less than 1%. But if we look at Klebsiella pneumoniae, we can see that in some countries, the resistance rate is higher. With Acinetobacter, resistance levels are higher yet, but if we look on a year-on-year changes since 2015, in E. coli and Klebsiella pneumoniae, resistance is gradually increasing, but within Acinetobacter, it's fairly static. And then finally, we've got Pseudomonas aeruginosa, and actually within the last few years, there's been a slight decrease in carbapenem resistance. So you can see, looking at the different countries, that there's large inter-country variations, with resistance being more common in Southern Europe than in Northern Europe. But this is just surveillance data. So these are isolates that are reported to ECDC, and these are just invasive isolates. And this probably doesn't really tell us the true picture of resistance within the different countries. And for example, within 2019, the number of CPE that were reported to the AMAHI reference unit from blood isolates was only about 8.9%. So if we look at this map of Europe, the countries are now coloured according to what's classed as the epidemiological situation of CPE. So this is where national experts within each European country were asked to stage their country based on whether they thought that there was sporadic occurrence of CPE right up to endemic occurrence. And you can see it's perhaps not surprising that within, for example, Greece and Italy, you can see that they're color coded quite dark. And this was the same within the ECDC IRSNet data. So that's not really surprising. But if you think back to the rest of Europe, most of those countries were colored in blue, meaning that the percentage of carbapenem resistance was less than 1%. And yet international experts within each country are judging their countries to have either outbreaks or regional spread of CPE occurring. So carbopenemases come in many different varieties, and we can see in red highlighted what we call the big 5 carbapenemase families. And these are the ones that account for most carbapenemase producers, not only in the UK, but globally. And as shown in this table, their distribution within the Enterobacterales and non-fermenters is quite variable. Although at least within the UK, the big 5 and combinations thereof account for the vast majority of CPE that are referred to the National Reference Lab, it's still worth bearing in mind these other carbapenemase families that are circulating, which are those that are in black text in this table. And just bear in mind these for isolates that you may encounter within the diagnostic laboratory that are negative for the big 5 carbapenemase families. So carbapenemase also differ in their global distribution, and this is shown in this map here. So within the UK, we have a fairly mixed epidemiology, mostly the big 5 carbapenemases, in particular OXA48 and NDM. But the situation differs in different countries. So for example, in the US and in Italy and Greece, it's KPC that dominates. In Turkey, it's OXA48 carbapenemase that dominates. NDM, most common in the Indian subcontinent. VIM in Greece and Italy again. And then finally, IMPs tend to predominate in the Far East. And having an understanding of this sort of global distribution of the different carbapenemase families can be quite helpful if you have a patient history. So for example, if you have a patient that's traveled recently to Italy, then you may suspect that they may have a KPC producer. So I'd just like to touch on now why we need to be able to detect carbapenemase producing gram-negative bacteria. So their prompt detection is essential for identifying infected and colonized patients because carbapenemase producing gram-negative bacteria are associated with significant morbidity and mortality. So it's important that their early and accurate detection takes place so that you can have rapid implementation of infection prevention and control procedures in order to prevent their onwards transmission. In 2020 in England, changes were made to the health protection notification regulations such that acquired carbapenemase producing gram-negative bacteria now form part of the list of notifiable organisms. So this means that any acquired carbapenemase producing gram-negative bacteria isolated from a human sample must be reported to PHE, and the results of any antimicrobial susceptibility test and any resistance mechanism identified must also be reported. And it's hoped that this information will help inform us on the epidemiology of CPE within England and also enable us to track the spread of outbreaks. So carbapenemase gene family detection can also inform patient management. And I've just summarized here how detection of the different carbapenemase families within gram-negative bacteria can help inform your choice of some of the newer anti-gram-negative agents that have become available in the last few years. So identification of the carbapenemase gene family is important for antibiotic stewardship, meaning that we can reserve these new antibiotics specifically for confirmed CPOs, carbapenemase-producing organisms, rather than using them for other organisms. And also the prompt detection of a carbapenemase family allows you to have quicker implementation of prescription of the right beta-lactam, beta-lactamase inhibitor combination for the right carbapenemase-producing organism. So now I'd like to go on to how we detect carbapenemase-producing gram-negative bacteria. And one of the main things to remember if you work in a diagnostic lab is that you don't actually need an acquired carbapenemase for there actually to be carbapenem resistance. So in some gram-negatives, carbapenem resistance is intrinsic, meaning that it's found in all examples of particular bacteria. And this is illustrated quite nicely by non-fermenters such as Acinetobacter and Pseudomonas, which are intrinsically resistant to ertapenem only. And in a seraceous species and the Proteae, we typically see poor susceptibility to imipenem only. So you see the imipenem MIC slightly raised. In some other gram-negative bacteria, such as Stenotrophomonas multifilia, Aeromonas species, and the Chrysiobacteria, these have intrinsic carbopenemases. So these are chromosomally encoded and again found in all examples of those bacteria genera. So in these, really what's key is accurate bacterial identification. And from then on, you can just assume that there's no acquired carbapenemase present in those isolates. In Acinetobacter baumannii, there's an OXA51 enzyme, which again is chromosomally encoded, but is only expressed if there are genomic rearrangements that lead to its expression. Most commonly in Pseudomonas, if you see resistance to just imipenem and or meropenem only and susceptibility to all other beta-lactams, then this is most likely due to OPRD porin loss and or efflux respectively, and you don't need to do any further workup on those. And then finally, within the Enterobacterales, if you have the presence of an ESBL or AMP-C together with cell membrane changes leading to impermeability, then you can also see the carbapenem MIC start to creep up. This is mostly observed in Enterobacter and Klebsiella and more rarely in E. coli. And it's in these latter organisms that we have the real problems with spotting carbapenemase producers. So in this schematic here, it's demonstrating how a true carbapenemase producer and Enterobacterales isolate that just has ESBL or porin loss resulting in carbapenem resistance. You can see similar MIC profiles for the carbapenems. And in fact, you end up with this sort of area between the two peaks there where it's not really possible to determine what the actual carbapenem resistance mechanism is. So in this case, it's important to remember that not all carbapenemase producers are resistant to the carbapenems, and not all carbapenem resistant organisms are carbapenemase producers. So you might think, well, what's sort of the underlying importance of this? Well, with ESBL and porin loss, or AMP-C and porin loss, the carbapenem resistance is not a transferable resistance mechanism, and it's also most likely associated with a fitness cost. So very rarely do these isolates go on to cause outbreaks, whereas we all know that CPE quite commonly, unfortunately, do cause outbreaks. So another issue is that carbapenemase detection is complicated by the diverse resistance profiles that carbapenemases confer. So all carbapenemases hydrolyze the penicillins, and in most cases, the cephalosporins, and to various degrees, the carbapenems, although they may have very low MIC values, and also aztreonam. However, some of the exceptions are OXA48-like carbapenemase producers that may appear fully susceptible to the cephalosporins unless there's an ESBL or AMP-C present. And similarly within beta-lactamase producers, unless there is an ESBL or AMP-C present, then these are typically susceptible to aztreonam. So you can see how the presence of other beta-lactamases, the level of expression of the carbapenemase, and the presence of other resistance mechanisms such as efflux and or impermeability can result in a range of resistance phenotypes. So I don't really want to spoil where I'm going with this talk, but the headline news really is that there's no universal method to readily detect and differentiate between all the different carbapenem resistance mechanisms. So if we start at the beginning, which carbapenem are you going to use for your initial screening? And UCAST have provided some guidance here where they recommend that meropenem offers the best compromise between sensitivity and specificity. So while ertapenem has increased sensitivity, it has a reduced specificity because ertapenem is most labile to ESBL or AMP-C with impermeability. And also you can't use ertapenem, of course, for the non-fermenters. Conversely, if you were to use imipenem, then it has increased specificity in comparison to the other carbapenems. But some carbopenемase producers, and in particular, some of the Enterobacterales can have quite low imipenem MICs. So the trade-off is that there's much reduced sensitivity if you use imipenem as your sole screening agent. And as I mentioned previously, carbopenemase producers cannot reliably be detected by clinical breakpoints. So carbopenemase producers can in fact appear susceptible if you just apply clinical breakpoints. So UCAST have defined screening cutoffs, which should be used, and I just need to emphasize here, for the Enterobacterales only. And the screening cutoffs are quite low. So for meropenem, it's an MIC greater than 0.125 and a screening cutoff of less than 28 millimeters. So UCAST have proposed a screening algorithm, and this is what they propose for the Enterobacterales. So you screen using meropenem as your indicator agent, and if you have a zone of inhibition less than 28 millimeters or an MIC for meropenem greater than 0.125 mg per liter, then the next step is that you go on to perform synergy testing. And as shown here, if you see synergy with boronic acid only, then this indicates likely a KPC or one of the other class A carbopenemases that may be circulating. If you see synergy between boronic acid and cloxacillin, then this indicates that there may be an AMP-C in your test organism rather than a class A carbopenamase. And if you see synergy with dipicolinic acid or with EDTA, then this indicates that there may be a metallocarbopenamase. So unfortunately, with the OXA48-like carbopenemases, there are currently no commercially available indicators. So the best indicator that we have at the moment for detection of these is resistance to temacillin. So as I said, this algorithm is for detection of Enterobacterales only. And the reason being, if you try to follow this algorithm for non-fermenters, then you're going to get yourself into a bit of a mess. First of all, the meropenem cutoff at greater than 0.125 is incredibly low. So most, I think, Pseudomonas and Acinetobacter will have meropenem MICs above that. And then when it comes to the synergy testing with Pseudomonas, you may very well see false positives for KPC activity, which is just due to de-repressed AMP-C activity. And both Pseudomonas aeruginosa and Acinetobacter are in fact resistant to temacillin naturally, so that doesn't indicate the presence of an OXA48. So for screening for acquired Carbopenemase-Producing Pseudomonas, you need to take a slightly different tack. And this is just a table that was from a paper that was recently published in JCM. And this was a study where they looked at a number of carbapenem-producing and carbopenem-resistant Pseudomonas aeruginosa isolates. And they found that if they looked at just resistance to imipenem and meropenem alone, then the sensitivity and specificity of using those two carbapenems as indicators of a carbopenemase producer, sensitivity was pretty high at 96%, so pretty good at identifying acquired carbopenemase producers. But as you can see, the specificity is very low at 36%. So essentially you're going to have a lot of Pseudomonas that are Carbopenem resistant due to OPRD loss and porin loss that are not acquired Carbopenemase producers. So a lot of additional follow-up work for not really much gain. However, at the bottom of the table, we can see the sensitivity and specificity if you include non-susceptibility to ceftolozane tazobactam, in addition to resistance to imipenem and or meropenem. And you can see that the sensitivity is now 100%. So using this combination, they were able to accurately detect all true acquired Carbopenemase producing Pseudomonas. With a little bit of a sort of trade-off on the specificity at 86%, but it's still much higher than 36% using imipenem and meropenem alone. And a similar study was also published last year by Gill and et al. As well, recommending use of ceftolozane tazobactam in addition to carbapenems for screening in Pseudomonas aeruginosa. Slightly different again in Acinetobacter species. So Carbopenem resistance in these is mainly due to OXA enzymes and typically most often OXA23, OXA40 and OXA58, but not OXA48, OXA58, but not OXA48. So OXA48 is vastly just limited to the Enterobacterales. And indeed, OXA23, OXA40, and OXA58 are pretty much exclusively found in Acinetobacter species. So these are quite tricky to detect in that there are no specific inhibitors for them. And many of the carbapenemase activity tests that I'm going to speak about next are quite unreliable for Acinetobacter. So the advice really is to report these as likely OXA producers without supplementary testing. And there's no requirement for you to refer these to AMOHI unless they need further workup, for example, typing or MIC determination for patient management. The one example I would say where that you would not follow that guidance would be if your patient has been hospitalized overseas recently, or if you do look for imipenem EDTA synergy in your lab and you do see that with an isolate, then in that case, it may be that you do have a metallo-beta-lactamase producer, and that's most likely to be NDM or IMP. So my recommendation then would be to screen either using a PCR or a lateral flow device, and also to refer it to AMOHI for further confirmation if it's from an invasive site. So I'm going to now go through some of the phenotypic methods that are available for carbapenemase detection. So starting with the modified Hodge test, and as I said at the top of my slide, it's very old, but it's not a good test really, and it's no longer recommended by UCOST or CLSI. So this method lacks sensitivity and specificity, and this is illustrated quite nicely by the little table here at the bottom right of my slide, where you can see that modified Hodge test was quite good at detecting KPC producers and also OXA48 producers, but pretty abysmal at detecting metallocarbapenemase producers. And you can also see that there were some false positives, with Carbopenem resistant Enterobacterales that were Carbopenemase negative. This quite commonly happens with ESBL or AMP-C producers with impermeability. It's quite a time-consuming test to set up, and it can be quite fiddly as well. And one of the additional problems is that it requires overnight incubation. So you're requiring a further 48 hours before you've got some sort of presumptive identification of carbapenemase activity. The results can be quite subjective. It can be quite difficult to read. And also it doesn't differentiate between the different classes of carbopenemases, so it's not particularly helpful from an epidemiological perspective. Probably the test that has been replacing the modified Hodge test more in laboratories now is the Carbopenemin activation method. And this is again another very straightforward test to set up, where you incubate a loop full of your test bacteria in a small amount of water or broth, and you add to that a meropenem disc and incubate that for 2 hours. You then take the disc out and place it on a Muller-Hinton plate, which has been seeded with ATCC25922, so a very susceptible E. coli strain. And you can then incubate the plate for 6 hours or overnight. And what you're looking for is whether there is a zone of inhibition around the meropenem disc following incubation. So if you have if your test organism is a carbapenemase producer, then as you can imagine, during that incubation step, the meropenem within the disc is degraded so that when you put it on the plate with the susceptible E. coli, there's no meropenem left and the susceptible E. coli can grow right up to the disc. However, if your test strain is negative, then the meropenem stays within the disc. And as you can see in the picture there, you expect to see a big zone of no growth around the meropenem disc following incubation with the ATCC25922 E. coli. So if you look in the literature, there's various versions of the SIM test, and the modified SIM and eSIM are recommended by CLSI. So the eSIM, just to mention briefly, is a further modification of the CIM test where you include EDTA, which inhibits metallocarbopenemases. So by doing that, it gives you an indication of whether your carbapenemase is also a metallocarbopenemase or not. Unfortunately, you do get some rare false positives. This seems to be particularly a problem with Enterobacter cloacae. And we've also noticed in the reference lab that you can get quite a high prevalence of false positives with Pseudomonas aeruginosa, although that does tend to depend on which version of the SIM test that you use. Again, it does require overnight incubation, and it's not recommended for use with Acinetobacter species. So what we're looking for with the next few tests that I'm going to talk about is they all have some way of monitoring for cleavage of the beta-lactam ring by the carbapenemase. So what happens is that there's a hydrolysis step and then decarboxylation. And as this reaction goes forward, what you see is the reduction in the local pH, and this can be picked up using a pH indicator such as phenol red or bromophenol blue. And then there's also a change in the mass between the indicator carbapenem molecule and the degradation products that are produced as part of the hydrolysis reaction. So I'll start by going through some of the colorimetric tests. So probably the best known ones are the CarbaNP and the CarbaBlue test. And these can either be set up using in-house methodology, or there are commercially available versions of these tests now. And then another commercial colorimetric test is the beta-carba test. This doesn't use pH indicator to detect the color change. This is actually a chromogenic substrate. So as it's hydrolyzed by the carbopenemases, you see this color change. And this is a commercial assay only. There's no in-house version of this. So the benefits of these tests in comparison to the phenotypic methods I've already mentioned so far is that they are quite rapid, so less than 2 hours till you get results. So you get results within the same day. And if you look in the literature, you see that they have very good reported sensitivity and specificity. There are, however, some issues with subjectivity of reading them. So although in these examples shown here, you see quite good differentiation between a positive and negative, you know, a very strong red versus a very light yellow color, and this is sort of same with the blues and the yellows in the blue carba test. But in reality, sometimes the results are a little bit kind of halfway house between the two, so it can be quite difficult to determine whether they're positive or not. And it is possible that you get these non-interpreters, which are probably due to weak hydrolysis, weak activity of the carbapenemase, leading to weak hydrolysis of the carbapenem antibiotic. Unfortunately, as well, we do see some false negatives, so it's particularly common with mucoid strains and also with some carbopenemases, and this is particularly noted with OXA48 and guest carbopenemases. So we can also detect hydrolysis using the MALDI-TOF, which I'm sure many of you are used to using now for bacterial identification. Using the MALDI-TOF for detection of hydrolysis of carbapenems is a bit different. So What you're doing with this is you take your test organism and you incubate it in a solution of carbapenem antibiotic for a couple of hours. And then you centrifuge your tube, and it's the supernatant that you are putting on your target plate, not the bacteria themselves. And then if you put them in the moldy trough, what you're looking for, as shown on the right-hand side here, you can see that we have certain peaks, which I've highlighted in pink, which correspond to the meropenem antibiotic. And when hydrolysis occurs, we see that there are some peaks that appear that have a lower mass than the meropenem antibiotic itself. So there are protocols that have applied the moldy trough testing to not only pure bacterial culture, but also it's possible to obtain bacterial cells direct from clinical specimens without culture, particularly from blood and urine, and perform this testing. If you look in the literature, there are many different sort of in-house protocols, and there's not really much standardization yet. So they may use different carbapenem antibiotics as indicators, and you also need to change your moldy trough mass detection range settings. So if in your lab your moldy trough is very busy, then it's going to take a bit of planning to kind of fit in your carbapenemase detection runs along with all your bacterial ID work. There are some issues with detecting some carbopenemases, and unfortunately, again, OXA48 crops up quite a lot. And then most recently, we have seen Bruker have produced a commercial kit that will detect carbapenemase hydrolysis. So that has some software as well, which helps you interpret your traces. So some of those synergy tests can be performed using different confirmation tests, and these are quite helpful because these will help you determine the carbapenemase class. So whether you've got a class A, such as KPC, a metallocarbapenemase, or by process of elimination, OXA48. So there's a variety of different methodologies, the most common ones being the double disc synergy test, which if you perform that, then using meropenem and imipenem is going to give you the best sensitivity and specificity, because some Enterobacterales are relatively susceptible to imipenem. You can get gradient strip tests which have imipenem and imipenem plus EDTA, which can be useful for detection of metallocarbapenemases. And again, because these use imipenem, they may not be so straightforward to read if you've got an Enterobacterales, because they tend to be more susceptible to imipenem. And then we've got combination disc sets, and then finally the Micro-0S broth microdilution plates, which use inhibitors along with meropenem. So just to summarize some of the benefits and caveats of phenotypic tests. So many of them are relatively cheap, and they're quite easy to perform. So just using antibiotic discs and sort of general lab equipment that you would have available to hand anyway. Many of them are also quite extensively validated because they've been around quite a long time. So these are generally the methods that are recommended by UCAS for laboratories that don't have much experience with beta-lactamase detection. Some of the caveats of them, however, is that they can lack sensitivity and specificity, and the results can be quite subjective. They can also be quite difficult to interpret if you have multiple resistance mechanisms present. So if you have multiple carbopenemases or an ESBL or AMPC in there as well, that can confuse things a little bit. Most of them detect carbopenемase production, but they won't actually tell you the specific families or the specific carbopenemase variants that you have. So again, you're going to lack information for epidemiological purposes. And most of them require pure bacterial growth. So that's already leading to a delay in you getting a result, and then also further overnight incubation. So now I'd like to go on to molecular methods. So there are many different molecular methods that are out there, which if you have people within your laboratory who have molecular expertise, then you can set up your own in-house methods. But probably the easiest thing to do is to implement some of the commercial molecular assays that are available. And there are very many commercial assays now available for detection of carbopenemases. These can be quite different. So they can either detect carbopenemases direct from pure bacterial growth, or we are starting to see more assays that are syndromic testing assays, in particular, mostly blood culture assays or isolates that will detect direct from respiratory samples. Generally, they have the same coverage, but I will touch on that again in a minute. The platform, it can either be a nice big expensive piece of equipment that you need to purchase specific to run that assay, or it may be a piece of equipment that can run many different molecular assays, or it may be that you can run the assay on a real-time PCR machine that you may already have in your laboratory. And they differ quite a bit in the hands-on time and the time till you have a result. So anywhere from between sort of 15, 20 minutes up until sort of 4 hours or more. So in terms of their coverage, they tend to cover the same carbapenemase families. But what's important to note is that the coverage within those families can vary between the kits. And this was highlighted quite nicely in an evaluation that we did some years now, where we looked at 450 pure bacterial cultures, mostly Enterobacterales, and we tested them against 4 different commercial assays that were available at the time. And in this table, we can just see the results summarized. So you can see for the KPC producers, the NDMs and the VIMs, the sensitivity and specificity of the test was excellent. We were able to detect all Enterobacterales that were producing these carbapenemase families. However, you can see with OXA48-like, we were only able to detect 83% of our OXA48-like producers. And this puzzled us a bit at the time. So we amplified up the OXA48-like genes and sequenced them. And we found that all of them were a variant called OXA181, which we now know to actually be the second most common OXA48-like carbapenemase circulating in the UK. So this was a bit concerning. So we fed this back to the commercial companies, and now those assays have been updated so that they do detect OXA181 variants in addition to other OXA48-like carbapenemases. If we look at the Caffeine Assay, it was the only one that said that it would detect imp carbopenemases. But even then, it wasn't able to detect all of the imp producers that we challenged the assay with. And again, when we sequenced the imp genes, we found that this was because the imp genes were not imp1-like. So the Caffeine assay only says that it will detect imp1-like carbopenemases. But if you are applying this assay particularly to Pseudomonas, then many of the imp variants are not imp1-like. So you will get, they're not true false negatives, but you won't detect the imp variant, which you may be able to do with other assays. So another molecular assay is the Immunochromatographic Tests, or the lateral flow devices, which I'm sure many of us are familiar with for sort of COVID testing these days. And these actually detect the carbapenemase enzymes. So there's a couple of different commercial versions of these available now, and they all promise to detect the big 5 carbapenemase families. And then the Chorus Resist series of immunochromatographic tests, there's also one that will detect OXA23. So this is the most common OXA carbapenemase that you tend to encounter in Acinetobacter species. So these are quite nice in that they give you results in less than 15 minutes, and they're quite sort of straightforward to set up. Just have to suspend a loopful of bacterial growth into a sample buffer tube, and then a couple of drops of that onto your immunochromatographic test cassette, and then you're looking for 2 lines if it's positive. The evaluations that have been published have shown that they have excellent sensitivity and specificity, not only from bacterial colonies, but also from some spiked clinical specimens. But it has been highlighted with some versions of these that you can get false negatives. We do see these occasionally referred to us in the National Reference Lab, and I think from experience, it tends to be more commonly NDM producers So that's just something to bear in mind. So the benefits of molecular assays is that they have much shorter turnaround time compared to culture-based susceptibility testing. And also there's the possibility of direct detection of carbopenемase producers from clinical specimens. So this obviously has improved treatment outcome for the patient, because the quicker you can identify a carbopenemase producer, then the quicker you can get your patient on appropriate antibiotic therapy. It also can lead to a reduction in empiric prescriptions. So if you don't have a carbopenemase producer, you don't necessarily have to go for the kind of big guns, the last line antibiotics, and that hopefully reduces some of the selection pressure that will lead to selection of the development of further antibiotic resistance. And of course, the quicker that you can identify a carbopenemase producer, then the faster that you can implement your infection prevention control measures. So many of these tests have high sensitivity and specificity, and in comparison to the phenotypic tests, they will identify the carbopenemase families, which is quite important for epidemiology. There's the possibility of increased automation, so minimizing potential human error, and also allowing batch testing, so releasing your laboratory staff time for other things. And it means also that carbopenemase detection is no longer a reference laboratory activity, so meaning that you can have localized testing and even near patient testing, which is obviously more beneficial for patient management than referring your isolates to the reference laboratory and the associated sort of 2 to 3 days or more delay in you having a result. Some of the things to bear in mind though with molecular assays, they're generally better to rule in patients. So any molecular assay is only able to detect the carbopenemases that it is designed to detect, which is a caveat sort of in comparison to phenotypic methods that just detect carbopenemase activity in general. So it means really that if you have a negative result, you can't reliably base your empiric therapy on that, because it may just be that the test bacterium has a carbopenemase that is not covered by the assay. And also the flip side of that is that detection of a carbopenemase does not necessarily be an accurate predictor of therapeutic failure. So there are reports in the literature of false resistance where you can have unexpressed carbopenemase genes or partial genes. And also implementation of or interpretation, sorry, of susceptibility based on a molecular test actually goes against the current UCAST and CLSI guidance, which just says that for patient management purposes, it's a MIC you need rather than detection of particular resistance mechanism. So I think at the moment, certainly for patient management, phenotypic susceptibility testing is still needed to guide patient treatment. One of the downsides of molecular detection as well is if you do direct detection from clinical specimens, then you risk losing epidemiological information. So it's still going to be important to culture whatever the carbopenemase producer is from your clinical specimen in order that you can undertake epidemiological typing for outbreak detection. And of course, there's the cost to consider as well. So some of the molecular assays can be quite expensive per isolate, but obviously you have to weigh that up against the cost of isolation of your patient for maybe 1 or 2 days while you undertake phenotypic susceptibility testing. So I just wanted to finish up by flagging this algorithm, which is taken by a paper by Workneb and colleagues, and it may not be that clear, I hope you can see, but I have sort of slightly amended it because the algorithm as was published was actually for phenotypic carbopenemase detection methods alone. But I think what the algorithm actually takes you through is also applicable to molecular assays or PCR, which is the only one that's not covered here. And I've sort of typed in where I think PCR fits as well. And it just talks you through really what you need to consider if you are looking to implement a method for carbopenemase detection in your laboratory. So obviously you need to consider the expectations of your service users, be that your clinicians or diagnostic labs that may refer isolates to you for testing. and sort of consider really what their expectations are. So do they want you to turn around a result very quickly? Does it have to be within the same sort of same day testing, or is it acceptable for there to be, you know, a result available the next day? You need to also consider what your local epidemiology is. So what carbopenemases are circulating in your local patient population. So it's no good implementing some of the assays that don't offer very good detection of imp carbopenemases if you know, for example, that within your patient population, imp carbopenemases are very frequently identified. Need to think about the cost of the platform and also the cost of the assay per isolate. And as I said on the previous slide, obviously any costs associated with implementing testing have to be weighed up against costs that are associated with isolating your patient and any additional costs associated with patient management while you're waiting for a result for carbapenemase confirmation. Need to consider if you need any additional equipment as well. So a lot of these tests don't require specialist equipment, but that is most likely a requirement if you want to implement a molecular test. And then thinking about also how the assay might fit within your laboratory's workflow, and of course within your laboratory staff expertise. So I just wanted to finish up by flagging these documents. So the one on the left here is the UCAS guidance for detection of resistance mechanisms of public health importance. So this is a really nice document. It covers not just gram negatives and carbapenemase detection, but also ESBLs and detection of resistance in other bacteria of public health importance. So for example, MRSA, vancomycin-resistant enterococci, and it's a really nicely written document. So I'd recommend you have a look at that if you haven't already, though it is getting a little bit out of date now, but the basics are still there. In the middle, we have the UK standards for microbiological investigation. So this gives you a really good overview of all the different molecular tests that are available, and this was updated as of last year. So another good document to have a look at. And then finally, we have the PHE commercial assays for detection of acquired carbopenemases. So this was a report that Danielle and myself wrote, and we're revising at the moment. So there will be a new version out fairly soon. And the purpose of this was to give the underlying evidence base for all the different molecular assays that have been commercialized. Really the aim sort of being that it's a document that diagnostic laboratories can read and look at the data within there and hopefully use that as a basis for selecting molecular assay for carbapenemase detection. So with that, I will finish. Thank you everybody for listening. If you'd like to contact us for any general inquiries, then I've put up the AMAHI reference unit email address there. You're welcome to contact us there. If you would like a copy of my slides from my talk today, then I've put my email address up there and you're welcome to contact me. And then finally, I'll put my Twitter handle up there as well. So thank you everybody for listening, and I'm happy to take any questions.
Fantastic presentation, Katie. Really valuable to see an overview of all the tests available to detect carbopenemases. So we currently have one question in the Q&A, but it's not too late. If you would like to pop a question in there, we can still get it answered. So just before we start the Q&A, I will briefly introduce our expert panelist, Dr. Danielle Munier, who also works at Public Health England in the Antimicrobial Resistance and Healthcare Associated Infections Reference Unit, where she is Deputy Head for the Antimicrobial Resistance and Mechanisms Service. So Danielle, if I could ask you to come off mute and turn your camera on, and I will hand over to you.
Good afternoon, and thank you to the organizer, and thank you, Katie, for the good talk. So yes, we've got the first question, and this is related to OXA48, which we know is quite difficult to detect, as you mentioned. So the question is, thymosilin resistance may not be seen not uncommonly in the Vitek antibiogram for Enterobacterales in the absence of carbapenem non-susceptibility, for example, in urines. Any advice for this? So what do you think? Should they be following up for OXA48? This will result in substantial amount of follow-up work.
Okay, I'm just having a read of the question myself to check that. So with you're saying that it's quite common to see timocillin resistance with the Vitek. Yeah, so timocillin, we do see within the reference lab timocillin resistance in the absence of OXA48. And there was actually an abstract at ECMID that was talking about timocillin resistance in the absence of carbopenemases. They didn't have an underlying resistance mechanism, but that was what they were investigating. It seems to be some sort of mutational resistance. How you would get around that with your Vitek testing. So presumably the Vitek has meropenem on it, though I don't know how low the concentration range goes. I think the Scottish AMR reference lab said that the meropenem MIC doesn't go as low as the UCAS screening cutoff, so they were just recommending whatever the lowest concentration was, and I think recommending doing a meropenem gradient strip test. But also, forgive me because I'm not that familiar with what's on the Vitek testing cards, but if you have amoxicillin clavulanate on there, then you would expect resistance to that with an OXA48 producer. I think in the UK SMI as well, they say that if you do not have a carbapenem on your first line antibiotic panel, then you should at least screen all amoxicillin clavulanate acid resistant isolates. So I don't know if that helps.
Can I just can you comment also, Katie, that some Serratia can be also resistant to timocillin?
Yeah, well, I think you've just said it there, Danielle. So we quite commonly get Serratia referred to us in the National Reference Lab because they are timocillin resistant, and timocillin resistance is not that uncommon in Serratia. So that's just something to bear in mind if you find that it's negative for OXA48 if you do go on to screen. I suppose in that instance, you would just have to be looking at the meropenem as well, because Serratia would be intrinsically resistant to amoxiclav, wouldn't it, Danielle?
Yes.
The ANSI.
Yes.
So they would be a little bit tricky.
Okay, thanks. So we've got a question, and I think this is close to our heart. So if the big 5 molecular test is negative, what should prompt lab to send for another uncommon mechanism to a reference lab?
Okay. So I think what we say in the UK SMI and the PHE CPE framework is that if you've ruled out the big 5, but your isolate has a meropenem MIC above the UCAS screening cutoff, or a zone diffusion size lower than the screening cutoff, to send it to us. The good news is if you make sure you write on the referral form that you have already screened for the big 5, then any further molecular testing that we do is free of charge.
So what would you say the importance of the rarer carbapenemase would be, Katie? Because most of this assay, as we know, focus on the big 5. Because as you said, the prevalence worldwide in the UK is for those are predominant, but what we say the importance of the rarer one will be? So
that's a good question. I think you need to have in the back of your mind awareness that you may have an isolate with one of those. We do have, if we look beyond the big 5 in the UK, probably the most common ones would be GES or IMI. Those would be most likely picked up by the phenotypic assays, though there are reports of them not being necessarily picked up. So, for example, GES carbapenemase can be a little bit tricky. We did get involved in outbreak investigation in a hospital where it was initially quite difficult to pick up the GES carbapenemase, and there was subsequently an outbreak because the first few cases were missed. One of the issues is whether these carbopenemases are really rare or it's just because people aren't detecting them. I think from the National Reference Lab perspective, because we routinely screen for, I think it's about 16 or 17 different carbapenemase families now, certainly we do have a good idea of how frequent they are amongst our referrals. And I would say it's not, unless there's a detection issue early on in the diagnostic laboratory with these sorts of carbapenemase producers, then they are genuinely rare rather than it being a molecular a detection problem. And some of the newer molecular assays are starting to go a bit beyond the big five, so there's also possibility of diagnostic labs looking for them soon.
Yes. So somebody thanked you for the very clear talk, and they said the Valabeni article was published in GCA. So I assume that CLSI standards have been applied to determine resistant versus susceptible versus non-susceptible. Does the table work with UCAS cutoff?
So I had a quick look at what the CLSI cutoffs were, and I think their cutoff for non-susceptibility was 4 mg per liter, which I think does tie up quite nicely with UCAS. And it also fits with what we do in the National Reference Lab. So with Pseudomonas, if we have a Pseudomonas with a caftolozane tazobactam MIC of 4 or greater, then we usually just pop that through for our PCR just to rule out carbopenemase. So I think it does still apply, yeah.
Okay. And there's a question about your thought on the best chromogenic plate for screening for CPE for patient samples, and I know what you're going to say.
I knew someone would say about screening plates. So I have to be honest, in the National Reference Lab, we don't have any experience with using chromogenic media for screening for carbopenemases. It's a bit difficult to say because if you read the literature, obviously different studies will say different plates are better, but you have to bear in mind what they've used for their verifications. And I suppose the only way you could truly assess which was the best plate would be to test again the same panel of carbopenemase producers against all plates. And I don't know whether such a study has been done. And as I say, unfortunately, it's not something that we've looked at in the reference lab. So I can't comment any further on that. There is a paragraph in the UK SMI, if you haven't already seen it, that does discuss chromogenic agars and some of the kind of problems there.
But just Katie, on our experience, I think we can comment of sometimes we get a lot of tests done on isolates with people starting with the plate, doing Vosco disc, for example, for the combination disc, and also doing after molecular or lateral flow. So where do you think people should stop at some point?
So I think in the UK SMI, there's a little flow chart, and it starts with clinical specimens, and it talks about direct plating on chromogenic agar. And I think if you get growth on the chromogenic screening agar, which is suggestive of a carbopenemase producer, it then indicates that you go on to do molecular testing of some variety, or it would then go into the pathway of standard susceptibility testing and then some form of molecular testing. So I think if you get growth on your chromogenic media, which suggests you have a CPE, I would probably, obviously I'm saying this without knowing from a diagnostic lab what your prevalence of getting things growing on it is. I apologize if you get things grow quite commonly on your chromogenic media that are not actually found to be carbopenemase producers. But I would say go with a molecular or immunochromatographic test from then, because otherwise it does seem that there's an awful lot of additional work being done with synergy tests and then immunochromatographic tests or molecular tests, and just sort of time's ticking on, and then you might refer it to us for confirmation of it being negative. And from a patient management perspective, that's probably less than ideal.
Okay, so we've got 3 questions. I think there's one we can answer quickly. I can even answer this. Hi, Kitty. Are there any available PTS EQS schemes for carbapenemase producing gram negative? So I think NEQAS have done a pilot for CPO detection, so maybe they will comment on that. And there's another one in Sweden. We have some strains of thymosilin sensitive OXA48 positive isolate. Do you think that we should, or you should change screening strategies?
Yeah, so that's something that's a bit concerning, is that there are more reports of OXA48 like positive isolates that are thymosilin susceptible. We've written a paper about them in the National Reference Lab, and there were a couple of papers at ECMID this year talking about it. I don't really know what UCAST are going to do and how they might change their recommendations. So I think it's a bit of watch this space, really. But it's worrying because these could be spreading undetected.
And the last question, what about commercial tests such as Resist5 or NG-Carba? Are they reliable?
Yeah, so that was one of my last slides, the immunochromatographic tests. I would say the published literature does suggest that they have good sensitivity and specificity. We've had a play with a couple of the different commercial tests, and our experience was that it detected pretty much all of the carbopenemase producers. The one thing that we found was possibly a bit of an issue with imp carbopenemases because they can be really quite variable. So it's quite difficult to design even a molecular or immunochromatographic test that will detect all the variants. So again, just be a little bit wary. You might get some false negatives, but generally very good.
And there's a last question or comment. Somebody has tested a panel of carbopenemases producer against several manufacturer plates as an in-house project and found that the EAO Carbasmart media to have been the best. So we can't really comment on this, but yeah.
But I suppose that's good to hear, considering that was kind of what I was suggesting needed to be done. So thank you, David, for sharing your experience there.
Okay, so thanks. I think we've gone through all the questions. Thank you, Katie.
Yes, thank you both. And just to touch on the EQA that we have, so we do have currently in pilot an EQA for Carbopenemases. We've sent one out. We're evaluating the results, and then we'll send out another 2 pilots and then evaluate before the scheme becomes live. So I hope you've all enjoyed today's webinar. Many thanks to our speaker, Dr. Katie Hopkins and Danielle for the Q&A round, and a big thank you to everyone listening for taking the time out to join us. We do have one more webinar left to deliver, which unfortunately had to be postponed due to reasons out of our control. And we are hoping to have a date for this very soon, and all updates will be made available on our website. In addition to this, UK NEQAS are also holding a series of digital and interpretive EQA pan-disciplinary webinars, which will be running every Monday in September. So for more information on those, please go to and visit the Central Office UK NEQAS website. So as with all our webinars, once we've closed this session, there will be a short feedback survey which will appear on your screen. Please do take the time and complete it for us, because your opinions do matter to us and we will be reviewing the feedback. So that's all from me. Thank you again once for joining us, and we will hopefully see you at the next webinar.